So good afternoon to all the participants and the panelists. Welcome to the third day post-lunch session of the event Recent Advances in Biosimilars Development. And today we have Dr. Satheeshkumar Nanjappan as a speaker. And as a brief introduction, I would like to introduce Dr. Satheeshkumar. He is a pharmacist and working as an assistant professor in the Department of Natural Products of NIPER, Kolkata. He has received his PhD from Jadavpur University, Kolkata. And Dr. Satheesh's research interests are focused on drug metabolism, pharmacokinetics, and drug transporters. In particular, his lab has been interested in identifying the possible herb-drug interactions, drug-drug interactions, and metabolite profiling. Dr. Satheesh is the principal investigator for research projects funded by various Indian government agencies. For his scientific achievements, he has received the Startup Research Grant, that is Young Scientist Grant by DSKT, Government of India, and awarded IDMA Young Pharmaceutical Analyst Award in 2014 for his outstanding contributions in the area of pharmaceutical analysis. And he was a recipient of IDMA Research Award 2015 and Indian Society of Mass Spectrometry Young Scientist Award in 2019. Dr. Satheesh has published more than 50 peer-reviewed research papers and 8 book chapters in various reputed journals and books. And with this brief introduction, I would like to invite Dr. Satheesh for his presentation on proteomics tools in characterization of biosimilar products. Over to you, Dr. Satheesh.
So, good afternoon, everybody. So, first of all, I would like to thank the organizer for giving this wonderful opportunity. Dr. Somasundram, so he is the organizer for this Untitled ACT program, and also I would like to thank Director Dr. Nanjappan for his invitation. So, as Dr. Somasundram here mentioned about the topic, the proteomics tools in characterization of biosimilar products. So I will be covering like some of the basic information so about what is proteomics, then what are the different kind of characterization tools available for proteomics. So how we can interpret those data with some additional inputs like bioinformatics tools, all those stuff. So we are going to discuss in this presentation. So Dr. Somo, how to I'm getting my screen, so the video is there. Can I able to minimize?
There is an arrow mark in the top. You can minimize it, and you go to your PPT.
Yeah. Okay. Now PPT is visible.
Yeah. If you click on the PPT, it will go to the next slide.
Okay. Thank you so much. So, as I mentioned, the topic is proteomics tools and characterization of biosimilar products. So, first, basically, we will discuss about what is proteome, right? So, proteome, proteins are biological molecules made up of building blocks of I will make so it is like biological molecules made up of building blocks called amino acids. The proteins are essential to life with structural, metabolic, transport, immune, signaling, and regulatory function among many other roles. So first of all, like if you see here, like the term proteomics, proteome was made by the Australian PhD students Mark Wilkins in 1994 in the symposium held in Italy. So it is a blanket term that refers to all of the proteins that an organism can express. Each species has its own unique proteome. Right? So unlike genomics will talk about the complete set of genes in each organism. Right? The composition of proteome is a constant state of flux over the time and throughout the organism. So therefore, when the scientist refers to the proteome, there are also sometimes referring to the proteome as a given point of time, such as embryo versus mature organism, or the proteome of a particular type cell or tissue within the organism. So now we have discussed about the proteome. So next we are going to discuss, so what is proteomics? So proteomics is the study of the proteome, investigating how different proteins interact with each other and the roles they play within the organism. Right. So although the protein expression can refer or inferred by the study of expression of mRNA, which is the middleman, we can say, like middleman between the gene and the protein. So mRNA expression level do not always correlate with the protein expression level. So further, the study of mRNA does not consider the protein post-translation modification, cleavage, then we can say complex formation, then localization, or the many variant of mRNA transcripts that can produce. All of which are the key to we can say the protein function. You can say the first experiment that fit the label of proteomics, the study was performed in, I can say, around 1975 with the development of 2D protein electrophoresis. So, however, truly the high throughput identification multiple proteins per samples only because of the possible with the development of mass spectrometry technologies for the 20 years. So since then the sensitivity, like we can say, if we talk about the proteomics, so then that too we are dealing with mass spectrometry. So since then we have to discuss about the sensitivity, accuracy of the mass spectrometry have made advancement to the point where the proteins can easily, we can readily detect down to the attomolar level. So that is called one target protein molecules per 10 to the power 18 molecules. So, and many other various other proteomics techniques also have been developed and optimized. So you can see here, we have discussed about the proteome, then proteomics. Then you can see here the current slide. So what are the key questions that proteomics can answer? So you can see here the broadly speaking, we can say that the proteomic research provide a global view of the process underlying health and disease cellular process at a protein level. To do to to do this, so each proteome, like we can say, each proteomic studies typically focus on one or more of the following aspects as a target organism proteome at the time to slowly build on the existing knowledge. We can say here you can see from protein identification to protein-protein interaction. So first is we can talk about, so what are the key questions that proteomics can answer is like the protein identification, then protein quantification, then protein localization, then is called post-translational modification, then functional proteomics, then structural proteomics, and protein-protein interaction. So first thing is we look into protein identification. So cell type each protein are normally expressed in a particular tissues or organ, as far as the protein are differently expressed. So if we talk about protein quantification, so how we can get the key questions to measure the total steady state of protein abundance, as well as investigating the rate of protein turnover. So that is how quickly the protein cycle between being produced and undergoing the degradation. So this process can be easily identified. So then again the protein localization. So here we can talk about where the protein is expressed and accumulates is just as crucial to protein function as the timing of expression, as cellular localization controls in molecular interaction partnership partners and targets are available. So then we come into the PTM. So this is called post-translational modification, can affect the protein activation, localization, then stability, interaction, and the signal transcription among other protein characteristics, thereby adding a significant layer of biological complexity. Then Going to the functional proteomics, this is the area where like proteomics is focused on identifying the biological functions of specific individual proteins, class of proteins, for example, like kinase or a whole protein interaction network. Then moving on to the structural proteomics, where you can discuss about the more insight into the protein functions like the drug availability of the protein target for drug discovery as well as like drug design. Right? So then the last one is like PPI, like protein-protein interaction. So investigate how the protein interact with each other, which proteins interact, and when and where they interact. So these are kind of data we can study when we are doing proteomic sort of analysis. So moving on to the next slide, so you can see like the whole overview of the different kind of proteomics tools are available, how the data we can generate, then how it is interpreted, all those stuffs are there. So first we can talk about the biological samples, so tissues, fluids, and cell lines. Then the proteins are peptide based on different sample preparations. Then the processed one we will put it into the proteomics tools to characterize those samples, right? So in this you can see like flow cytometry is there, then mass cytometry, then mass spec, then 2D page, then protein microarray. So if you look into it, like most common proteomics tools involving mass spectrometry, as I mentioned, these are the most important tools available when we talk about the proteomics. Right? So in this, because of this extensive data produced by the proteomics tools, so how we will interpret those data? So that is very like we can say it is an airplane task. So for that we can see here is what are the data generated by this tool that should be incorporated or it should be taken as support by bioinformatic process that is called like database and analysis. So in this, so functional analysis, then protein assembly, then protein or peptide identification, then biomarker discovery. So because as I mentioned, so when we do sampling by using proteomics, so the number of data will be there. So we have to process, we have to characterize those things with, then obviously we have to take some support like bioinformatics tool, like in silico tools or database and analysis help to characterize those data. So that is really it is necessary for the analysis of hundreds of molecules. So next you can see once it is done, so you can see like the assessment of candidate biomarker through like analytical as well as you can see risk clinical validation, right? So if you talk about like analytical validation, so here it is like they involves the use of different methods. So for an example, you can say like as molecular level, then immunological, then proteomics. You say in molecular level, the RT-PCR is there. The immunological is instant blot, ELISA, then QT page. Then the final one is like proteomics. So some of the tools we have discussed here is like cytometer, then mass spec and mass cytometer. All those tools are there to get the data for characterization. Then followed by you can see like clinical validation. Here you can see like the retrospective or prospective studies for like diagnosis, prognosis, and disease monitoring and forecast treatment. Right. So this is the overall tool when we talk about the proteomics, how the sample has been taken, then those samples, how it is used for like in the proteomics aspect. Then after that, so generated data, so how we are interpreting with like bioinformatics tool. So then followed by the like fingerprinting, all those things will come. Right. So these are the overview. So when we talk about, then another thing is like we have discussed flow cytometry, then mass, protein, microarray, then UV page, all those things. Apart from this, you can see like mass cytometry. This is like the mass cytometry is an next generation flow cytometry platform, which we can say like which utilize uh, like elemental mass spectrometry to detect the metal conjugated antibodies that are bound to intracellular or extracellular to antigens of interest on single cell. So that is the important thing of mass cytometry. So here you can see the number of cellular parameters that can be simultaneously monitored by conventional fluorescence flow cytometry assay is inherently limited because of, like we can say, like inherently limited because of fluorophore emission spectra overlap. So this is the limitation. You can select fluorophore emission spectra overlap. So in contrast, we can say, however, the mass cytometry accurately discriminates the metal isotopes of different atom masses without any channel overlap. So that there we will get the clear-cut information. So that is the advantage of like mass cytometry. Then you can see here is like this is the platform of a mass spec with the common merits that enable the comprehensive protein characterization. So here you can see like contemporary mass platforms like ion traps, linear ion traps, then FTICR, then triple quad, time of flight, then RB trap. So all those platforms are there. So in this, so how we are getting based on the contemporary mass platform. So what are the advantages? What are the things we are getting in terms of like characteristics? So if you look into the right hand side, you can see the merit, biggest of merits. Like based on this mass spec tools, you can see like the selection based on the resolution, like then product ion mass accuracy. Another most important thing is you can see like the linear dynamic range, then MSN capabilities, then reaction conditions, accessible between the stages of mass, then ionization, ionizer compatibility, efficiency, then automation. All these steps are there. Like because if you see the improvements in proteomics instrumentation over, you can say like 5 to 10 years in concomitance with the improvement of chromatographic or electrophoretic separation, the mass spectrometer, you can say like sensitivities and resolution have led to more precise characterization of physicochemical features, as well as we can say like impurities in biosimilar preparation. So in particular, we can say like the mass spectrometry based structural proteomics tools are capable of performing rapid, then parallel and large scale characterization of proteins, and thereby defining identity, we can say like then quantity, structure, and function of biotherapeutics can be done based on these are the advantages I'm saying, particularly on advancement in the mass spectrum, right? Important tools we can say like all having we can gone through about the rapid, parallel, and large scale characterization, all those things. Apart from that, so under this category include mass spec like coupled with various separation methods. As I mentioned earlier, it is with chromatographic or electrophoretic for protein identification and protein complex deconvolution. So label-free techniques for protein quantification and analysis of interaction, kinetics aspects, and gel-based methods for intuitive profiling, all those things. So these are the advantages. A number of different innovation is happening in respective to mass spectrometry. Based on this, so we can able to characterize the of physical properties of any kind of samples. So here you can see like the role of mass spectrometry at different stages on clinical part respective to biosimilar. So you can see here is throughout the development of biosimilars. So mass spectrometry has different function at different stages, right? So that you have to agree from. You can see here is from characterization to verification of biosimilarity, that is to analytical comparability. So if you see from different stages, you can see there is like development starts with detail, you can say like structural characterization of the innovative product, where the mass spectrometry already plays a crucial role. You might be knowing, like with the involvement, we can able to easily characterize or analyze the data, whatever we are doing in the proteomics part. So according to, we can say, like respective to European Union and US regulatory requirement, the amino acid sequence of biosimilar product must be exactly same as that of the innovator. Therefore, the determination of amino acid sequencing is the most critical part of the initial characterization. So there, based on these regulatory bodies. So these are the things, as I mentioned, the sequencing is the most important part of the biosimilar product. So it should be exactly same as the innovator, right? So there you have to keep in mind. So whenever we are doing this sort of analysis, so by use of mass spectrometry tool, so we can able to easily do the characterization. The complete structural characterization of the innovative product may serve as the starting point for shaping the, like we can say, QDPP, like the quality target product profile, which is a specific way should be defined for the product under development. So complementing this on the basis of risk assessment is also involved to determine the critical quality attributes. So that is called those structural features of the active substances that may have influenced its efficacy, then safety and immunogenicity profile. Right? So that all the things you have to keep in mind. That what each and everything from characterization of the product, innovative products, there it will deals with, like you can say, like the sequencing, determination, then characterization of glycolysis, then determination of disulfide bridge. So then identification of microvariants like non-enzymatic post-translation modifications, then disulfide mismatching in sequence variants. All this stuff. Then if you move into like molecular genetic transfections like and cloning and clone selection. So you can see the verification of amino acid sequence, cloning the sequence variant, then again characterization of glycosylation, then confirmation of disulfide bridge. Then moving to the process development scale of production, there you can see like batch to batch comparison. This is most important thing. So always that should be like batch to batch comparison. So how it is deviating from another batch. So this based on it, as I mentioned, like the critical quality attributes, monitoring via peptide mapping or by intact mass measurements, all those steps we can able to derive the endpoint. So then further structural characterization of standards and clinical batches, then identification of again process related impurities of, like we can say, like post cell protein, all these stuff that also we are going to discuss in the upcoming slides. Then again the important thing is verification of biosimilarity, like analytical comparability. You can see here is detailed structural comparison of innovators and biosimilar batches, like injectants of units, mass analysis, comparison of major isoforms, then peptide mapping, then comparison of you can say like high order structure via like hydrogen deuterium exchange mass measurement. So each and every tool, it is complementary to each other. We can I usually disable my video because I think I'm using the input problem. Dr. Somu, Dr. Somu, yeah, my voice is audible.
Yeah, it's audible now.
Okay, okay, because you went back, I think I lost my connection because
Yeah,
of
yeah.
it. So, yeah, so we are discussing about like the role of mass spectrometry at different stages of the non-clinical part of biosimilar. So, in continuation to that, I think the diversity of the biosimilars, as I'm I think a couple of days back, for the 3 days, people are saying the same thing. Like, so in US FDA, how the biosimilar is called, then European Union, how the biosimilar is called, then WHO, how it is called. So all those steps, just I get for information. So I'll go through it like USFDA. It is like a biosimilar. Uh, it's a biological product that is highly similar to and has no clinical meaningful differences from an existing FDA approved reference product. So in European Union, like the biosimilar is a biological medicine, so highly similar to another already approved biological medicine. So in WHO, like similar biotherapeutic products, a biotherapeutic product that is similar in terms of quality, safety, and efficacy to an already licensed reference biotherapeutic product. So because we are discussing like based on the different regulatory agencies, how the biosimilars are classified. Right? So as you know, like biosimilars are not generics and are the copies of already approved products that have been proven to be a similar to the original products. That is called reference product. So apart from this, you must know like there is called biobetters or bio superior. So it's an additional term which has often been used to when we discussing about the bios. So biobetters and bio superiors. So this is the additional terminology. So we have to keep in mind. So again to the term of generic, we can say like these terms are not applicable to biosimilar as the word biosimilar is a regulatory term which has a specified shortened pathway for approval. So that is called biobetters or biosuperior. The term biobetter or superior we can say are not indicative for any regulatory pathway, but a marketing team for classification of new or second generation products. So typically we can say biobetter the molecules may be similar to the currently approved product, but the molecules are usually altered to improve the attributes such as we can say like the changes in the dosage regimen, then we can say like improved safety, then efficacy or immunogenicity. So because of these significant differences in the molecular structures and molecular functions, so biobetters are considered as like new biological entities and would follow the standard approval pathway to get then regulated, set by the regulatory agencies. So for an example, we can say here is like um, pegflagrestin, so which provides an improved half-life and dosing regimen when compared to the pegflagrestin. Right. So this is some of the example, like if we talk about the biobetters and biosupri. Then next is like you can see, then again this is more some of the things might be repetitive. So we can say like the reference for biological development timeline and cost. So what are the phases from discovery to end phases? What are the things it should pass? Then if we talk about the biosimilar development time and cost, so what are the from regulate development phase to the phase three? So how much time, how much year it will take place, all those stuff it is there. I'm not going to um, elaborate more because these are the differences. I think hope you might be heard a lot about this. So next you can see like another important thing is like the critical quality attributes. So somewhere we have discussed about the critical quality attributes from you say here the role of mass spectrometry at different stages for the non-clinical part for biosimilars. So there we have discussed about the process development and the scale up production, all those things. So there we have discussed about some of the parameters of critical quality attributes. So then we have to see what is critical quality attributes. It is in the physical, chemical, biological, microbiological property are characteristic that should be within the appropriate limit, range, distribution to ensure the desired product quality. So, how we can get this critical quality attributes? For that, we can use quality by design. So, this is the quality by design is a systematic approaches to development that begins with predefined objective and emphasizes the product and process understanding and process control based on the sound science and quality risk management. If you can able to see like the ICH guidelines, so there is having like ICH QER2 that is for pharmaceutical development. Then Q9 is called quality risk management. Then then ICH Q6B, it is for specifically it will talk about the specifications of biological products. So in this, so it is like systematic approach to development that begins with predefined objectives. So based on this, so we can get a predefined objectives. Based on that, the thorough process understanding and process control with sound knowledge, so we can minimize the or target the critical quality attributes so that we can assess and we can move on to the endpoint analysis. So that is called like you can see is QTPP. So like selection of attributes, different attributes, then criticality of assessing, then how we are like statistically how we are doing our plan for similarity assessment. So how we are using this biosimilar process development. So how based on that, how the analytical testing and non-clinical testing will be done. So how this data endpoint will be analyzed. Then if there is any issues in this, then how we can refine these parameters. So all those stuff we can do in by using quality by design parameters. So moving on to the next is like you can say like this is the basic mass workflow for the protein characterization, right? So here you can see like from start to end like biotherapeutics, how we can like enzymatic treatment, then followed by like final thing is like biotherapeutic. fingerprint. So how we are getting this finite endpoint. So from starting to the biotherapeutics, we can say like characterization begins by like we can say like the proteolytic digestion of the therapeutic protein followed by different steps as we mentioned like chromatographic or electrophoretic separation, right, of the compounds. Then you can see like top one is we can see left hand side mass spec analysis will give an overall picture of any possible variation or modification in the protein structure. Then it is move on to the MS2 mass spec 2, the spectra which is acquired after subjecting the peptide to fragmentation using like we can say like novel functionalities. So attached to the mass spectrometer such as electron captured dissociation or like pollution induced dissociation. So based on this, the database search of the acquired spectrum facilitate in deriving the protein fingerprint. So here you can see what are the changes. So from start to like proteolytic digestion, then how the chromatography techniques we are using, then based on the mass spec, then all those things, then after generating the data, then based on the database, as we mentioned in the like a couple of slides back, where we use the bioinformatics tools to pick like database sets like picking. So then specifically Mascot we can use, then Swisspot database too. So based on this, so we can derive a biotherapeutic fingerprint. So because as you know, like mass spectrometry gain prominence in protein characterization with the development of soft ionization techniques such as like the basic thing is like the spray ionization. So and MALDI are there. These are the two prime soft ionization techniques. So ESI and MALDI, right? So these tools made more like mass spectrometry more amenable to analyze large molecules which was hitherto limited to smaller compounds due to difficulty in transferring the former into charged gaseous phase. Right. So though MALDI is suited for protein characterization, the associated constraints with regard to the analysis of complex protein flexibility and you can say like charge state produced by MALDI makes PSI as a more advantageous tool for characterization of biotherapeutics. So this is a quite evident we can say from the number of like publication you can see in the proteomics analysis for biosimilars. So you can get a number of scientific literatures in respective to this areas for characterization of proteins with a single amino acid residues, resolution, and so on. So this will be, we can say, it will therefore not be an exaggeration to claim that mass spec-based characterization has become the most highly utilized analytical techniques for characterization of biotherapeutics due to its versatility. Then important thing is like sensitivity and the ability, like we can say, like ability to decipher both small and large alteration in biologicals such as sequence variants, then diversity in glycosylation profiles, then other we can say PTM like post-translational modification, including sulfide shuffling and so on. So this is the overview of the structural characterization of biologicals, how from start to end, so what are the things are involved, so how we are getting the data, the picking, then how we are incorporating the correct bioinformatics tool to get the endpoint data. So that is more important. So apart from that, you can see is the highlighted portion, it is there here. So the scope for further improvements in this sample preparation like enzymatic treatment, there we can make some in this area we can have a chance for the improvement. Then again the fragmentation pattern like ion mobility, then hydrogen deuterium exchange mass. So again, then next is like bioinformatics, picking all those stuff. So really here we can like more innovative approaches we can adapt to get a current endpoint analysis that we can see. So next is you can see here is like the different approaches for analysis of proteins or proteomes. So you might be knowing like if you see in the right hand side, so there is four approaches are there, right? So from you see the last one, fourth one, you can see like top down proteomics. Then next is the middle down, then extended bottom up, then bottom up proteomics. So you can see is like the list structural information will be given in like top down proteomics. Then if you go higher side up to like bottom up proteomics, so here you can get more than 99% of sequence confirmation, almost complete protein fingerprinting will be get by this bottom-up proteomics approaches. So you can see like difference, the top-down approach and bottom-up approach. You can see like first is like protein extraction, then protein separation will be followed by then mask detection and analysis. So if we talk about like bottom-up approach, we can say in the proteomics. So it is also termed as like shotgun proteomics. So all the proteins in the samples are first digested here in a complex mixture, right? So of the peptides, and the peptides are then analyzed to identify which proteins are present in the sample. So that is the difference if we talk about the top-down and bottom-up approach. So another important thing is like you can see apart from this, so here the top-down proteomics more than if you see the peptide size more than 15 kilodalton. So here we can use it can be analyzed. Then here the future is like a confirmation of elemental composition and nature of modifications. So this kind of features we will be getting if we are using like top-down proteomics. Then the middle down, so you can see the peptide size is 7 to 15 kDa. So more complete sequencing information, the nature and location of more modifications. Then extended bottom-up approach proteomics, there you can use the peptide sizes around 3 to 7 kDa. So near complete sequence confirmation, nature and location of most modification, then disulfide linkage, glycosylation and profiling, all those stuff you can get in this approach. So the bottom-up approach, as I mentioned, it's a shotgun proteomics. So you can see is like peptide size is around 0.6 to 3, like mostly like 99% of sequence confirmation and at most complete fingerprinting you will be getting, right? So another thing is you have to note it down like top down and bottom up we have discussed. So another thing is like middle down approach also you can see this approach is an emerging approach. One which has the potential for we can say the successful application in future for the study of isolated or purified protein as well as the protein. So this has been devised and introduced recently based on merits and demerits experience in the bottom up and top down studies. So that's the future. We can say like the bottom up and top down approaches has been combined in an appropriate manner with the objective to arrive optimum conditions that constitute the middle term approach or promising. We can say like this implies uh, that approach is also involved in the study of terminated peptides, like instead of like intact proteins obtained by the proteolysis or chemical degradation step, which is like characterized by bottom-up approach. But the size of, as I mentioned earlier, but the size of the resulting peptide would be greater than the one that we are using encountered in bottom-up approach. Right? So the things respective to different approaches for the analysis of proteomes or proteins. So moving on to the next is like you can say like the peptide mapping. So you can see. So this is one of the important uh, assessment for primary sequencing assessment, and one of the most regulatory requirement for biosimilar is to have identical amino acid sequencing as the reference product. If the farmer differ from the latter in terms of other microgenetics such as the different in you can say PTMs like post transition modification provided all those stuff. But here you can see like the mapping. So what are the different steps are here? Like once it is digesting from the protein, so how you will digestion will be happens? What are the different steps? So you can see here is like these are the different chemical reagents can be used for digestion of the protein. When the protein digestion by step, which is involved in 5-step process, you can see here like the denature of the protein, then alkylate, then quench, then trypsin digestion, then lower digestion pH to all the trypsin activity. So based on the sample preparation process, so we can quench then the same things we can be done through mass spectrometry. This is one of the things, important things. So protein mapping, then protein mapping, then you can see here is like apart from the journal. So here in this peptide mapping, so another advantage I forget to incorporate here is like this technique is used for like apart from that like batch to batch variation during the bioprocess development. There, it is most like we can say comprehensively we are using to check the preliminary screening of batch to batch variation during bioprocess development. The constant, we can say that constant evolution of proteomics instrumentation has advanced the techniques of peptide mapping considerably because of the advancement and innovation in mass spectrometry. As an example, we can say like data independent acquisition, like if we talk about like peptide mapping approach, so with the alternate low and elevated, we can say like pollution induced ionization energy screening. It can be investigated difference in the protein sequencing between the biosimilar candidate and corresponding innovator product too. So this approach also, like we can say, allowed for identification of mutations in heavy chain of the biosimilar. The the main observed difference we can check in peptide mapping according to uh, the many of the research articles published so far. So it can be perhaps to be attributed to inconsistent integrity of the expression clone, which if corrected at an early stage of biosimilar development could be significantly reduce the chance. We can say significantly reduce the chance for the product failure in the later stages of the product development. That is the advantages if we are doing like in the initial stages. This is the uniqueness of the method. So in this we can like take like we can say like fragmentation of the precursor ion was performed independently of the prior to the precursor ion scan. Then after that characteristic features of the data independent analysis based on the analysis for like you can say different sort of approaches we have check here like top down, bottom up, and middle approaches, all these stuff, right? So these are the things. The limitation is we can say like when we talk about like protein, sorry, peptide mapping is which in the later is that like detectability in like a dynamic range is restricted to the peptides that ionize the base. So, however, we can say the method can be allowed accurately 5 over the wide dynamic ranges. When we used about like uh, short ranges, all those things, so detectability will be uh, questionable. So, but the thing is, we can able to make like accurately quantification over the wide dynamic range. So this would be like more advantageous. So next we can see like another tool is like native mass spectrometry. So you can see native mass spectrometry is increasingly being used in probing structural biology of therapeutic drugs, including complex monoclonal antibodies, conjugates, targeting multiple molecular targets or mechanism of action. So you can see like
So, Dr.
the low
Satish,
sample requirement with hello, yes,
yeah, there's some
yes,
background. I think somebody speak.
somebody is
You can.
speaking. Yeah, okay.
Okay.
Now it is clear. Yeah, okay.
Yeah. Now it's.
Okay. So yeah, if you talk about like the native mass spectrometry, so low sample requirement with the ability to monitor the dynamics and the subpopulation in the sample. Same sample being it is probed is a characteristic feature of the native mass spectrometry that overcome the limitation of we can say the traditional tools like X-rays and NMR, so which require flexible part of protein to be removed for the analysis. But here it is like we can make a probe kind of setup so that we can do the analysis, right? So some of the limitation, as I mentioned here, is like X-ray and NMR tool. So there they require the flexible parts of the protein to be removed for the analysis. So since apart from that, we can say like since the key objective of the biosimilar characterization is to determine any structural changes that impact the clinical performance of the drug. If it is of prime importance that the analytical techniques used allows for retention of dynamism of the molecule. That is called, we can say, the functional native conformation without damaging, you can say, damaging their covalent and non-covalent interactions without damaging. So that is which makes, we can say, which makes this measurement more promising. You can say that is the use of native mass spectrometry. So while we can, another thing is we can say on ESI top mass spectrometry continues to be used for the intact mass measurement. The user of new version like arbitrary mass spec, then other have allowed biosimilar characterization workflow with the multi-fold enhanced resolution that has been enabled identification of multiple co-occurring post-translational modification and other modifications. So we can say like in recent the publication also providing a number of detailed data about comprehensive protocols for intact mass analysis of different maps modalities with the recommendation of about applications, how we can use the time of flight analyzer versus other that may require some more higher resolution version of the mass spec. So these are still, it is like more innovation is happening in terms of to get more predictable endpoint analysis. So next one is more another important and interesting thing is uh, ion mobility mass spectrometry. So there you can see this is we can say it is a prerequisite to obtain optimal ionization for efficient mass analysis. The biggest concern we can say is if we talk about the ion mobility mass spectrometer when we performing the protein analysis in any mass format is to retain the solution phase structure in gas phase. So several researchers have reported the occurrence of changes in the protein structure upon transitions from solution to ionized gas phase. So addition to this advantages, addition to these advantages, we can say like ion mobility spectrometry, we can to the mass spectrometry offers an attractive solution and solution to this problem by allowing the separation of ions, that is allowing the separation of ions that you have to keep in mind. So allowing the separation of ion, like we can say isomers, then isobars, then conformers that have an identical elemental composition but different only in the structural arrangement based on the differential mobility, right? So there we can able to use this advantages when we use this mobility mass spec. So the larger molecules have like the higher pollution across the section, like smaller molecules, thereby we can transversing in different times in chamber filled with net gas under the influence of electric field. So here you can see like a pictorial view of the ion mobility uh, mass spec. So it is like electric field, then drift region is there throughout this, then drift ring, then ion shutter, then ionization source, then all those stuff here. The important thing is like you can say like the transferring in the different times in the chamber filled with inert gas under the influence of electric field. There is the three common variant of ion mobility spectrometry are commonly used in protein characterization. So most important thing is like we can say like three is like drift tube as is shown here is drift tube in this pictorial picture. So that is called, we can say it is drift tube or a DT type tube. So differential mobility analyzer, that is also called like DMA. Then the final one is like traveling wave, that is called T wave type instrument. So ion mobility even more attractive in the possibility to couple ion mobility spectrometer to multiple MS variants. We can say like such as like time of flight or linear protocol or some kind of like trapping devices or FTIR like Fourier transform ion cyclotron resonance and magnetic sector spectrometers. So thereby like we can say like harnessing the advantage of each and adapting the analysis according to the need, and we can get the questions whatever we require or the questions to be answered by based on this kind of uh, innovations in ion mobility spectrometer. So that is the advantage as I mentioned, like it can be often the attractive solution to the problem by allowing the separation of ions, like we can be able to differentiate isomers, isobars, and conformers. Right? So next would be glycosylation. So again, so this is again one of the important aspect if we talk about the critical quality attributes due to the influence of biological activity, then efficiency and stability, then immunogenicity, bioavailability, like and ADCC, that is called antibody dependent cellular cytotoxicity, and complementary dependent cytotoxicity for the commercial therapeutic glycoprotein. Here you can see based on this kind of analysis, you can see here is why we need a glycolation analysis and how to control the system for manufacturing glycoprotein products. Here you can see like it is like the complexity as I mentioned earlier. This is having the complexity of the structure, then site specificity, then heterogeneity, then the impact on efficiency and safety, then comparability, then the time consuming for optimization, then the final one we can say is high skill requirement for the analysis. Obviously, like if we talk about the proteomics too, that too, excuse me, if we are talking about like biosimilar characterization. So obviously, like we should have high skill. So because n number of data we used to get, so how all the data can be taken into consideration, how we are going to characterize those data, then how we will get into the endpoint analysis, the final outcome. So all those things are matters like complexity of the structure to high skill requirement. So all the things matters when we talk about the glycosylation analysis. So another thing we can see from the perspective, like in terms of biosimilar development, it is not even possible to have identical glycosylation profile as an inhibitor. It is very difficult. However, the profiles need to be similar, and the regulatory approval requires that are different to be justified with regard to the clinical impact. We can say like these consideration under the necessity of monitoring glycosylation pattern for biotherapeutic drug during all stages from start to end. We can say like all stages of manufacturing as to ensure the desirable product quality, then batch to batch consistency and the product stability, all those stuffs. Like complete structural registration of the glycan requires the determinant of the sugar composition and sequencing, like sugar sequencing, like the monosaccharide branching, then interglycosides linkage, anomerasin confirmation. So leveraging on all the things like whatever we have discussed for glycolation, batch to batch, consistency, all those stuff. So improvement in fragmentation techniques, it can be like we can say like earlier also we have discussed like CID and UTD, like pollution induced dissociation, then the electron transfer dissociation, then charge reduced species, all those stuff derived from to gain more information about the site of glycosylation modification as well as the sequencing of the peptide fragments. So this all stuff will be added advantage when we talk about the confirmation of like desired product quality and batch to batch consistency and the product stability. So next is like assessment of high order structures, like this is the determination of protein conformation is an important component of comparability studies for biosimilars. Particularly we can say like instance where the conformation changes occurs without any changes in the primary structures or sequencing. So often we can say these changes are triggered by initiation of degradation pathways that rendered the protein immunogenic by aggregate formation. So here it is like the changes or modifications or alterations at the primary level of protein can be detected or characterized with tandem mass spec LC mass approaches. However, for assessing the changes that results due to the degradation of protein, as we discussing about the assessment of higher order structures, it should be like more about the degradation of proteins. So requires probing of higher order structure. So how we can be able to check this endpoint analysis respective to the degradation of protein. So we can say by using like assessment of high order structures like pairing of SDX like hydrogen deuterium exchange mass spec is being adapted in the industry with high degree of successes. We can say like when we talk about the degradation of proteins, right? So HDX we can use. It has been like instrumental for providing detailed insight about like a PTM post-translation modification, then conformational changes, and more importantly, we can say like in comparability studies of biosimilars and innovators. If you use high order of like structures, these are the technique is based on like we can say HDX if we talk about. So this technique is mostly based on the differential uptake of deuterium in exchange of protons by different conformation of biomolecules, right? So that is the main thing if we talk about the HDX. The another thing is important thing we can say like differential exchange rate corresponding to the different conformation is captured by mass spec as a deuterium, like it has been like higher mass compared to the like hydrogen, so which get reflected on the acquired mass spectrum, right? So apart from that, we can say like HDX is also used for like uh, epitope mapping of protein therapeutics. Then HDX, MX, particularly you can say for biosimilar development, including examination of like the effect of different types of glycosylation on maps, confirmation and protein receptor binding, then confirmation and characterization of the changes in the variants of human monoclonal antibody and so on. So we can say finally, like if we talk about the assessment of high order structure, so this application with HDX is again, it is like innovation is happening, still it is continuing and to be adapted more areas in biotherapeutic analysis. So next is you can see like assessment of host cell protein. So this is the host cell protein. You can see like the complexity of the different process that are used to manufacture the protein therapeutics along with the raw materials that can be result in co-purification of some of the uh, host cell proteins that bears like close resemblance of the protein of interest. Broadly, we can say from here it is as we discussed like clone selection, then media automation, then bio reactor control, then after that like some sort of like uh, um, processing. Then 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 there is like we can say three platforms are there majorly to monitor the host cell protein. So here you can see here is first one is uh, ELISA based host cell profiling, protein profiling. Then next is like gel electrophoresis based host cell profiling. Then the final one is you can see here is mass spec proteomics based host cell protein profiling. Right. So these are the three platforms are used for monitoring HCP. So the first one, if we talk about like ELISA based HCP profiling, like the method helps to quantify the amount of host cell protein present, but do not provide the details about the identity of unwanted proteins. So that is the lacuna if we talk about the ELISA based thing. Platform. Then next you can see is gel electrophoresis based profiling. So here is this assay we can talk about. We can talk about they have higher coverage than the a previous one, that is ELISA. So like ELISA, but they have lower dynamic range, right? So next one is like proteomic based profiling. So mass spec based proteomic platform enables detailed characterization of biotherapeutics, like both you can say like qualitatively and quantitatively with the dynamic range much higher than the these two platforms, as I mentioned, the ELISA based platform and the gel electrophoresis platform. So, the host cell proteins, and another thing is like the host cell DNA are produced by microbial or mammalian cell are being used for the expression of the product of interest, right? So, the most of the things, so we talk about like the processes, the most of these the impurities we have to talk about this process, then again we have to talk about the impurities. Most of these impurities should be removed in the early stage of separation, and their presence in the final products occur, it should be at very low levels, right? So basically, as I discussed in the couple of slides back also, so if you talk about the liquid propylene from or like product development start to end. So the early stage only, so we have to sort it out by using like innovation techniques we have to use, and we have to quantify all the like degradants or impurities, so that if you go into the end scale, like end of the process, so that you won't face any problem, because of the impurity levels also might be very less. Right, so these are the advantages I'm talking about. So till now we can say the ELISA-based method are predominantly used by the industry for wholesale protein quantification in various processes like intermediates and final products and so on. So, however, as I mentioned earlier, like there is a key limitation if you talk about the other platforms like ELISA. Like one of the things is like limitation for ELISA is like inability to detect the weak immunoreactive proteins. Then and also like to define the identity of wholesale protein. So this has fueled a lot of efforts towards finding the better analytical approaches for HCP profiling. So taking together, we can say like the outcome of the extensive HCP profiling studies, it can be easily concluded like that the platform of the impact of HCP have on safety, then efficacy, well characterized wholesale protein profiles can aid in devising the efficient, we can say like devising efficient development, like product development, and subsequently the purification strategies that can we boost up the overall the process of bioprocess productivity. Right? So, so major we can say another thing is advantages like advancement to have been made in our ability to measure, how to characterize, so how to remove the contaminations, like as we talk about the impurities in the biotherapeutic process. So each and every process should be thoroughly as I mentioned, like we can uh, uh, take an guide from QBD approach, like quality by design approach. So from start to end, so each and every process will be thoroughly monitored so that we can minimize like degradations and impurities, all those stuff. So moving on to the next is like biosimilar development chain. So here you can see like these are the development chain, highlight the functional characterization, like functional characterization, like the functional characterization is a routine undertaking from the discovery processes for measurement of consistency between the batch to batch. So variations here you can see like from defined target, then product development, then followed by the consistency between the batch to batch. So to check the batch to batch variations, then manufacturing. If you talk about the batch to batch variations, like the physical chemical characterization, then biological characterization, right? So these are the steps are there. From this, like how to like a different cell-based assays, how we can get like what are the assays we can use, like a cell-based assays, binding assays. So what are the in vitro assay tools are available when immunogenicity assay. So if we talk about like the histochemical characterization, like comparability, then stability, as we have discussed in previous slides too, like comparability studies, then stability studies, then preclinic and clinic, all those stuff. Like while if you talk about like the cell-based assays are the most common platform for the potency assessment and the in vitro binding assays using label-free techniques. Again, we can say like surface plasma resonance and bilayer uh, interferometry are gaining popularity because as they allow the detection and monitoring and characterization of the biomolecular interactions. These are the ability uh, uh, to cope up with this mass spectrometry tool to give the possibility of proteomic based functional characterization studies for biotherapeutics. So apart from that, in this like biolayer interferometry assay systems are there. This is again, it is a label-free techniques for the measuring biomolecular interactions. So it is an optical analytical techniques that analyze the interferences pattern of white light reflected from the two surfaces, like we can see like the layer of immobilized protein on the biosensor tip and the internal references. Layer of surface plasmon resonance technology have also been used for antibody characterization and quantification. So next you can see like different characterization of biotherapeutics using the proteomics tool. So you can see what are the like the proteomics tool, particularly the mass spec. Different mass specs are used for characterization of biotherapeutics like LC mass, small d, then FTMS, then different approaches, small d, then capillary electrophoresis, then hydrogen deuterium exchange mass spec. So are used for the characterization of biotherapeutics in the proteome as a proteomics tools. Again, here some again more mass spec tools are there for glycosylation and host cell protein profiling. So these are the snapshot we can say like how we can use different mass spec tools for characterization of proteomics. So next you can see here is like the commonly used software package for quantitative performance. So it has been categorized if we can say like label based, label free, and integrated platform. So three categories have been there: label based, label free, and integrated platform. So if we talk about the label based software package for quantitative proteomics is isobaric, Q, then eye tracker, Libra, then protein pilot from ABSIX, then from Thermo you can see Proteome Discoverer, then view, then Express. So these are the tools can be used for like different sort of like relative quantification of the software that can be used for like isobaric tag of for relative and absolute quantification with tandem mass tag and labeling also. Then if you talk about the label-free techniques, then again EM5 we can use, and then Muscat distiller, then Max LFQ, then Viper is there, then you can use for integrated platform and specific tools like OpenMS, then Peak Studio Express, then Skyline, the commonly used software package for proteomics quantification. Then next is you can see like the different computational tools available for, we have discussed about the bottom up, middle up, and top down approaches. So in this, we can see like the combination of all these three approaches. You can see these are the computational tool like we can use Mascat, Sequest, Mamsa, then Extandem, Post PC, Mass Shoot, Yada, then MS Allen Plus. So this tool can be used for all the approaches. When we are going for specifically for like bottom up approach, we can use like mass base, vinyl, then Morpheus, peak, and sensors and so on. So if we specifically if you are using only like middle down approach, then XDA computational tools are available, we can use. Then if you want to go for specifically for top-down approaches, you can use Topic, then Mass Pathfinder, then ProSite, then TopFind, and so on. So these are the computational tools. When we talk about like the characterization of proteomics and so on, so we can use this tool to get the endpoint analysis. So I hope I have reached the summary. Yeah. So I would say like the proteomics is an indispensable tool in the characterization of biosimilar, like the expansion of biosimilar market around the globe. We can say we primarily depends on the addition of more sophisticated platform, like we can say like to the analytical armory that can be exploited for characterization of biologics. So based on the development, what we have discussed in the current presentation, so we can say that mass spectrometry based proteomic approach are and will continue to be significant enabler in this regard. So then while we have discussed about some of the existing tools that have been successfully used for qualitative and quantitative measurement of protein structures and function, right? So that we can far up from obtaining the complete fingerprinting of bio therapeutics. So this is all from my side. So yeah, thank you so much.
Yeah, thank you, Dr. Satheesh, for your detailed presentation on various applications of these proteomics tools in characterization of biosimilars as well as various biological products and its applications. Thank you very much.
Thank you.
And I request audience any questions. I think no questions. If the audience have any questions, you can also post it by mail or even, yeah. So just your feedback, insightful presentation, sir. In a glance, you presented the C, like given a detailed explanation about proteomics. Yeah, thanks for that suggestion or comment. And yeah, if any more questions, you can always post those things by email or the WhatsApp group, and we will get back to you with the answers. Yeah, thank you. Thank you, Dr. Satheesh, for accepting this invitation and providing an excellent talk on proteomic applications. So thank you. And for the next session, we will meet tomorrow morning at 10 a.m. Thank you all.